In this last lecture on the cytoskeleton, I will be discussing on cell motility. So many cells actually are capable of moving by themselves. So not just unicellular organisms like amoeba, or those with flagella and cilia like paramecium and dinoflagellates, but even cells in the multicellular organisms like your white blood cells or mesenchyme cells in embryos. So cell motility that we will be talking about here is mainly focused on cell movement that doesn't involve flagella nor cilia. So if you want to know more about that, you might want to go back to the previous video on cilia and flagella. So here we'll be focusing on cellular motility based on actin filaments. So the actin filaments in a cell would actually allow it to be able to change its shape as it moves. So as we can see here, this cell is actually moving towards the right. So that's the direction that it's moving towards. How do we know that? Because of the formation of a thinner, more spread portion in this tip of the cell, as opposed to a sharper and thicker tail that we see here. Okay, so this is the leading edge of the cell. So here is a cartoon view of that movement. So that broad and thin leading edge is actually known as a lamellipodium. So it thins a bit as it extends. And the reason why it is able to extend is because of the actin filaments that are forming. And here we can also see the integrins playing its part. So they hold onto the surface as this portion extends. Okay, so there's an animation. So you can see the movement of the integrins in sync with the change in the shapes of the cell. So this lamellipodium that we see here is actually quite a general thing in moving cells, especially cells that don't have flagella nor cilia. And we can also see other projections from the lamellipodium. Okay. And these projections, as I mentioned a while ago, are caused by the formation of new actin filaments in this portion of the cell. So there was an experiment on attractants. So attractants are chemicals that would attract cells to move towards it. Okay. So this cell has been attracted to move towards this direction. And what's happening here at the molecular level? So we can see here the lamellipodium forming. And this is the tail end. So notice here in this chemical reaction that new actin branches, actin filament branches are actually formed. Okay, so these purple blobs that we see here, okay, actually is the ARP2-3 complex that would actually give rise to a new branch of actin, as we see here. So as new actin grows, it would actually sort of push the cell membrane also out of shape. So that's why the lamellipodium has this weird shape, because it's the actin filaments that are growing and pushing it that are causing this change in shape. And those actin filaments that have already grown their maximum length are capped with capping proteins, and this is to protect them from degrading in this particular end. Okay, so new branches form, as we see here. Here's a smaller new branch, and this will actually extend on and on as long as the cell needs to do so. While on the opposite ends of the actin filaments, proteins will also help in deconstructing the actin monomers because they're not needed anymore as filaments. So again, that's because of the different actin binding domains. Okay, so these actin binding domains like this for end blocking or capping, for depolymerization as we see here, for filament severing, and for filament nucleating, the purple blobs that we saw a while ago, are important in cell movement with the use of actin. So here's a picture to even prove that one. So we can see here various hierarchy of Y-shaped structures. And those yellow blobs that we see there are actually the ARP2-3 complexes for nucleating actin filaments. And the blue-green, green, and orange and gold-colored branches that we see here are actually growing actin filaments. And in this particular picture, the actin filaments are color-coded differentially from the myosin filaments. And we can see here their positions relative to each other. Actin filaments are sort of positioned all throughout the cell that's trying to move towards the direction that the arrow is pointing. So you can see here the lamellipodium, and this is the tail end. So the actin filaments are distributed all throughout, while the myosin filaments are sort of concentrated here in the middle towards the tail of the cell. And why is that? Because the myosin filaments are still going to pull the tail forward. Okay? So can you imagine a really fat blob trying to move forward? It will move first its front part, the lamellipodium, and later on it needs to pull everything with it. Okay? So, and that's being done by the myosin as we see here. And we can also stress map. So when we say stress map, I'm not talking about psychological stress. I'm talking about material stress throughout the cell. We can see here that the greatest of the stress are actually somewhere here in the center. Okay? Where the myosin are exerting effort so that they can pull the tail towards the direction of where the cell is moving. And we can also see here an interplay of, guess what these attachments are? Those are integrins, and you have the actin filaments. Okay? So if you can recall what this one is, then you better recall it in your mind right now. So we see the actin filaments are not just anchored into nothingness. They are anchored onto integrins. Okay, so remember that. And there are actin-dependent processes during development, such as the formation of the branches of the neurons. Okay, the certain neurons in your body are able to move where they're supposed to go. So the connection of neurons. Okay, so the axon needs to migrate from one point to another in order to connect certain parts of your body, to innervate them. And we can see here these sharp projections are actually actin formed filaments that are going to sense the environment. We can see here the actin filaments. And the rigid structure that we see here in the middle are actually microtubules. So here's a time-lapse photo of a neuron that's being attracted towards a particular target cell. So you can see how this could be the axon, grow towards the target location. And actin filaments also help in the changing in the shapes of certain cells. So if you can remember on our lesson on cadherins, that it's the cadherin that causes cells to actually be grouped together with their similar cells during embryonic development. Actin also plays a role there in terms of reshaping the cell depending on how they are arranged. So notice here from fairly square-shaped cells, the cells become sort of columnar and as they become part of the neural plate. And the change in the shape of the cell is actually due to the presence of microfilaments or the actin filaments, as we see here. So it sort of serves as the thread. If you look at the seams of your clothes, if you look at the thread, the actin filaments here are acting that way so that the cells sort of are modified in shape, as we see here. So there's a curling here because of the change in the shape of the cell brought about by actin filaments. And also here. So this is just a picture of that neural groove that's forming in an embryo, brought about by the elongation, by the way, of the cells are brought about by microtubules, but then the changes in shape of the cell, they become wedge-shaped, is due to actin filaments. And then there are equivalents of cytoskeletons in prokaryotes. So we have this one, the PAR-M molecule, which aids in the separation of genetic material during cell division in prokaryotes. And we have the CRES molecule, which aids in the change in shape of certain cells. So here we have a comma-shaped cell, and that is dictated by the changes in shape of the CRES protein. And we also have the FTSZ protein, which aids in sort of the cytokinesis between these two newly formed bacterial cells.